Alrighty, so we're going to try this out. Welcome to meiosis. Before we start, let's talk a little bit about chromosomes, just as a reminder. When you see a line here, kind of looks like a V, that is one chromosome. We could also call that a chromatid. During interphase, the first stage of meiosis, this chromosome will replicate itself, and it will look like this. We also call that one chromosome. I know that's a little confusing. However, it's not just a chromosome, it is a replicated chromosome. So this half of the chromosome here is the same exact copy as this half here. However, the reason why we call it one chromosome is because we typically measure chromosomes by centromere. So here would be one centromere. Up here, this is where the centromere would connect. So they're both considered to be one chromosome. However, this is a replicated chromosome. There are two chromatids here, 1, 2. We call those sister chromatids. All right, so let's move on to it. So we start out meiosis with interphase. There's only one interphase in this entire process. During interphase, the cell is doing its normal cell things, like dehydration synthesis, osmosis, diffusion, hydrolysis. The cell is growing in size. In addition to this, the cell is going to replicate its DNA. So in here, we see that this cell has 1, 2, 3, 4 chromosomes. We're going to duplicate those chromosomes. And we still have 1, 2, 3, 4 chromosomes. But if you count the chromatids, we have 1, 2, 3, 4, 5, 6, 7, 8 chromatids. So we duplicated our number of chromatids that are there. So we went from 4 to 8. So this is what interphase looks like after replication. And now we're ready for our first round of PMAT. Prophase 1. We have normal things happening that would happen in a prophase. The spindle is forming, the centrioles have migrated, the asters are anchoring everything into place, and the nucleus has disappeared. We are now able to see our chromosomes within the cytoplasm. In addition, in prophase 1, we have the forming of tetrads. So our homologous chromosomes are going to find their counterpart. Remember, for every chromosome you get from mom, you get a corresponding chromosome from dad, the same types of genes on them. So we have to have those line up together and we form a tetrad. This is one tetrad. This is another tetrad. We call it a tetrad because every tetrad has 4 chromatids within it. There's 1, 2, 3, 4. 1, 2, 3, 4. The prefix tetra means 4. In addition to the formation of tetrads in prophase 1, we also have a process of crossing over, and that is where tetrads will literally switch information with each other so that we now have 4 completely genetically different chromatids. Here we have a yellow, a blue-yellow, a yellow-blue, and a blue. Down here we have a green, a purple-green, green-purple, and purple. So all 4 of these chromatids and all 4 of these chromatids are completely genetically different. So prophase 1, to recap, we have all the normal things happening in prophase. We have the formation of tetrads, where the homologous chromosomes get together, and we have crossing over. The purpose of crossing over is to create more genetic diversity. In metaphase 1, the tetrads will line up across the middle of the cell, so we don't have that single file line that you would expect. Instead, our couples, our tetrads here, line up across the center. In anaphase 1, the tetrads are being pulled apart. So they were in their pairs, their homologous pairs here in the center. We pull one side of the tetrad one way and the other side of the tetrad the other way. Same thing with these. In telophase 1 in cytokinesis, our centrioles are gone, our spindle is gone, and we now see the formation of two nuclei because this cell is going to split into two. From here, we immediately go into prophase 2. We do not go through another interphase. So right from telophase 1 in cytokinesis into prophase 2. So obviously now we have two cells that were created in our last stage. So in prophase 2, it's not as exciting as prophase 1, what we have going on here. The spindle is forming, the centrioles have migrated, the nuclei are gone, and we now see our chromosomes floating in the cytoplasm. In metaphase 2, our chromosomes are going to line up in the center and get ready for division. The spindle connects to the centromere of the chromosomes. In anaphase 2, the chromosomes are pulled apart into individual chromatids and pulled to opposite sides of the cell. In telophase 2, we have the spindle is gone now, the centrioles are gone, and we have two nuclei forming in each of our cells, here and here. These will now split down into our final product, which are 4 cells. 1, 2, 3, 4. These cells have half the amount of DNA, so we started with 4 chromosomes. We now have 1, 2, 2, 2, and 2. So we have 2 chromosomes in each of our cells, which is half of the amount that we started with. These 4 cells are gametes, which means they are sex cells. They are haploid, meaning they have half the amount of DNA that we started with. They are all also genetically different from each other and different from what you started with. So if we look at how they're different from each other, we have a yellow and a green, yellow, blue, green, purple, blue, yellow, purple, green. Remember, order matters. And then we have a blue and a purple. So they are all genetically different from each other and genetically different from where we started. There are 2 types of meiosis. You have meiosis in males that will make sperm and meiosis in females that will make egg. Every time meiosis happens in males, we get 4 sperm. Every time meiosis happens in females, we get 1 egg and 3 polar bodies, which cannot be fertilized. Hopefully this helps. If you have any questions, see me. Have a great day.